Okay, dear participants, we will begin the seminar in about 1 or 2 minutes. In the meantime, we are allowing a few more minutes for everyone to join. A quick reminder, please make sure your Zoom display name matches your full name as used during your registration, first name plus last name. And by joining this session, you are considered to have accepted the data privacy consent as part of your registration. During this session, you may ask your questions by either unmuting yourself and speaking directly or by using the Q&A or chat sections. Thank you for your participation, and we will get started shortly. Gjorgji, you prefer to take questions during your presentation or after your presentation?
For me, it's the same. If there is questions in the audience, yes, I can answer directly the question, but they will need to speak immediately because I will not be able to see any raised hands and everything.
Okay, thank you.
Just please stop me and I will try to address the question.
Thank you. We'll begin in 1 minute, okay? Okay, let's start. Dear participants, before we begin today's session, we would like to take a moment to express our deepest condolences for the recent passing of Dr. Antun Balaz, our colleague from NCC Serbia. Dr. Balaz was a passionate researcher, a dedicated member of EuroCC community, and an invaluable contributor to the advancement of high-performance computing in the Southeast Europe. His legacy will continue to inspire our collaborative efforts across the region. Our thoughts are with his family, friends, and colleagues. Today's seminar is dedicated to his memory. At this time, we would like to kindly invite some of our colleagues from NCC Serbia, Maria and Dusan from NCC Serbia, as I mentioned, should they wish to say a few words in his memory. Dusan?
Yeah, unfortunately, today I must share that our dear colleague and close friend, Dr. Anton Balázs, died unexpectedly on Friday at age 52 in Szeged, Hungary, and this is shocking and tragic news. We are all deeply devastated, and we will miss him very much. So Anton was a dedicated physicist and supporter of science in general. He was also a respected mentor across generations. He was also my mentor and the closest collaborator in both research and infrastructure projects. His contribution made him a prominent figure in Serbia physics community, and his loss is deeply felt by the Institute of Physics Belgrade and Serbia research community as a whole. Here at the Institute, he served as a deputy director. He was also a member of Serbian Academy of Science and Arts, and here at the Institute, he led the Center for the Study of Complex System and the Scientific Computing Laboratory. He was also a member of Optical Society of Serbia, European Physics Society, and the German Physics Society. Anton's research was focused on ultra-cold quantum gases, especially Bose-Einstein condensation, as well as numerical simulation of complex system, Monte Carlo simulation, high-performance computing, and functional formalism in quantum theory. So in these areas, he led many national and international projects. He published top scientific journals, and he was author of influential textbooks and monographs. Antun was also member and leader of many regional projects, including SIGRID, HPC, VSIM, NIFOS Europe, and others. So he was very committed to international collaboration. He was especially dedicated to working with young researchers and was closely involved for decades with Petnica Research Center Station, which is oriented to work with young talents here in Serbia. And in Petnica, he also served as vice president and of the board of the directors. His legacy remains. His ideas, courage, and achievements continue to motivate us and will steer our future. May he rest in peace and be remembered with honor.
Thank you, Dusan. Maria, do you have anything to add or say something about Balas?
Sorry.
It's hard, I know.
Yeah, I think Dusan said everything. Thanks.
Thank you. Following this short remembrance, we will proceed with our seminar as planned. We thank both our speaker and our participants for allowing us the time to honor Dr. Balas at the beginning of today's session. Thank you. Now we welcome you to the fifth session of the 5 Beats of Intelligence AI Meets Diverse Domains seminar series organized under the EuroCC-4C project. Today's seminar is hosted by EuroCC-Turkey and contributed by EuroCC North Macedonia, and we are honored to continue exploring how artificial intelligence intersects with diverse scientific domains. It's our great pleasure to introduce today's speaker, Professor Gjorgji Madjarov, from the Faculty of Computer Science and Engineering at SS Cyril and Methodius University in Skopje, I guess. Gjorgji is a leading researcher in the development of advanced AI methods for analyzing complex multiomics datasets. With over 17 years of experience in research, development, and leadership, his expertise spans time series modeling, stream processing, and AI operations. He currently leads a team of top professionals delivering innovative machine learning solutions worldwide. Now we are excited to hear his insights on how artificial intelligence is reshaping the landscape of genetic research and its application. Gjorgji, now thank you again for joining us today, and the floor is yours.
Thank you very much, Suzanne. Thank you very much for this opportunity to give a talk regarding the emerging trends of AI within genetics, but not only in genetics, it's within bioinformatics in general, and analyzing and building models for multiomics data and multimodal data. So I'm going to share my screen right now.
Please.
Yeah. Can you see my screen?
Yes, perfectly.
Yeah, good. So, like I said, I'm going to share some insights regarding the new trends and emerging technologies within bioinformatics, especially in analyzing genetic and epigenetic data, but also something that is very important for the transcriptomics and proteomics. We are witnesses today that we have an explosion of generating different type of data, especially in the biomedical domain, which means that we are generating a lot of data, but our tools are not prepared to analyze autonomously to some extent the data that we are generating. Also, we have a huge problem of generating the data as a vast amounts, but actually we are making generation of multiple patients that are under some investigation or some particular research that we are performing. So we are dealing with high dimensional data, but with really small number of examples that we are trying to analyze. If we put everything together, until now we have somehow focused of analyzing the data to some certain extent in the scope of the project that we have or the scope of the research that we would like to perform. But the scale of that analysis and modeling is far away from what we would like to achieve. And we can also witness the big potential of the new technologies that have been developed in natural language processing, which means that right now we are living in a time where the large language models or the models that are produced within this domain demonstrate great potential and great capacities, and we would like to learn something from that domain. So what we would like to perform is actually how to basically transfer that knowledge that we have acquired with the large language model and how to transfer everything within the domain of bioinformatics and analyzing this data that could be from different sources and different quality and different species, for example. So I would like to introduce here how these new technologies completely transforms the current landscape of machine learning and artificial intelligence that performs within this domain. And I would like to share some recent studies regarding building of foundation models that are able to digest and process billions of different data points, but also different data modalities in order to acquire some additional knowledge for the domain that is specific to our research. So from what we have seen, and we are pioneering with this kind of a concept in the recent years, like 3, 4, 5 years ago, but recently in the last 6 months to 1 year, we have seen an explosion of different type of models that are somehow part of this ecosystem of foundation models that enables us to better understand the whole domain that we are working. And I would like to share some details regarding the foundation model that have been recently built, on what kind of task they have been learned, with what kind of data they have been learned, but also what are their advances if we take into account and compare with the traditional machine learning models and artificial models that we have seen in the previews that were related to the bioinformatics domain. And of course, because like I said, we are generating all the time huge amount of data, but not all the data have a same data source and the same modality. The biggest advantage here is that we can process and digest different types of data, and we can acquire multiple different knowledge from the different data representation that we can put in the models. The thing is that we would like to somehow build some kind of models that will be able to generalize well across different tasks, to be able to acquire some additional knowledge. And once we have a specific problem or specific, we are saying downstream task, we would like easily to fine-tune that model for the particular task that we are interested in. So the first thing is that we need to acquire and digest a huge amount of data, and from that data to grasp the patterns and the knowledge that exists hidden within those data. And afterwards, we would like to perform some kind of a fine-tuning of those models in order to be able to solve some specific task. And that task could be from different nature, predicting some health condition for a patient, or even or predicting some states, or what is the influence of specific drugs, or the specific drug that the patient receives, and etc. So before we start explaining this concept and what kind of data have been used for training this foundation model that are very important right now in the field of bioinformatics, I would like just to remind our knowledge and refresh our knowledge regarding the type of data that we are using here, and what are the core concepts that we would need to grasp before we start to build such a model like I'm going to explain. So basically, from the human perspective, we have 22 pairs of chromosomes plus one pair of chromosomes for the sex chromosome. Within males, we have XY, and for females, we have XX. And the chromosome is basically this part of the human genetics. And basically, within the chromosome, we have it is basically composed of DNA and the proteins on the other side. The main representatives of the proteins are the histones here that we can see on the picture, that are basically 8 molecules, two molecules from the there we have basically 4 different types of molecules, which are basically 2 of every of them. So in total 8 different molecules: H2A, H2B, H3, and H4 histone proteins. And for us, it's very important that these histone proteins are highly positively charged proteins, which means that the DNA on the other side and that kind of a sequence of nucleotides, which is represented by 4 different nucleotides: A, C, T, and G, adenine, guanine, cytosine, and thymine. Basically, they are highly negative charged, which means that based on those charges, they try to interact between each other in order to condense the DNA structure basically into one into the chromosome and into the nucleus directly. So what does it mean? That means that actually our DNA, if we make some kind of a sequencing, we can observe the sequence of nucleotides within our DNA, and our DNA locations. But instead of a sequence, one linear dimension, actually our DNA is represented in the 3D space, and basically some locations that are far away from the other, they could be very near in the space, which means that they could be somehow close within the 3D space representation. So basically, with the 1D representation of the DNA, we lose a huge amount of information in order to understand how the different parts of the DNA can interact and influence on the other parts of the DNA that we are observing. The most important thing here is that once we have this molecule here, there is a cycle across those molecules from the DNA. Two cycles basically makes. And the thing is that actually they have some kind of interaction, and if that interaction between the histone proteins and the DNA is relaxed, that means that some kind of enzymes, in particular polymerase enzyme, will be able to start transcribing of the specific DNA that is open and relaxed and has a relaxed interaction with the histone proteins. So what does it mean? That if this here part of the DNA has a relaxed interaction with the histone proteins, we can have we are enabling basically the enzyme polymerase to get inside and to start the transcription of the genes that are particularly part of that location within the DNA. And basically the first thing is that what we obtain there is the pre-mRNA, which is premature RNA, and then that premature RNA start to the splicing there, and we obtain only the genes or the parts of the gene where the exons are exons take part of that transcription. And all the introns that we have within the DNA, within the genes, they are just removed from there, and only that part of the exons is transcribed and then continues to synthesize the proteins. So for us, it will be very important not to get insights only for the genetic part of our DNA, but also we are interested in about the how strong is the interaction basically between the proteins here and also the DNA. Here between the nucleosomes, these combinations of the DNA and the histones, we have some kind of a linkage protein in these positions here in order to be able to basically condense and tightly cupped package the DNA in order to be able to be part of the to be settled within the nucleus. So the important thing is that we need to figure out that when the gene expression is performed, it means that in those locations we have some kind of a relaxed interaction between the proteins and the DNA. So we can influence that interaction and how strong is that relation by modifying, for example, the proteins and also modifying the DNA structure, which means that with additional of specific molecules, we can basically influence on the relaxation or exposition or suppression of those of some particular genes. And for example, if we have acetyl group on the histone proteins, it means that that part of the DNA will be completely relaxed, and the RNA polymerase can basically start the transcription of the DNA. If we add, for example, one particular methyl group for the to the histone proteins, also the interaction between the histone proteins and the DNA will be relaxed, and we can allow the gene expression. But on the other side, if we add multiple methyl groups to the histone proteins, it means that actually that interaction will be tied up, and basically the RNA polymerase cannot enter in the DNA structure, and it cannot make a transcription of that part of the DNA, or the gene cannot be expressed, we are thinking. Why this is very important? Because this gene expression or the regulation of which part and which genes are expressed are part of the epigenetics, which means that we would like also to identify some kind of a different markers that will tell us more about which parts or which genes will be expressed and how that will influence to the synthesization of the proteins within our body in order to keep a healthy metabolism, for example, in our body. So there are different markers that we are sensing across the DNA while we are sequencing the data, but we are interested in additional markers, not only the sequence of DNA. So within the DNA, we can have some kind of mutations, and we can see those mutations by analyzing directly the DNA sequence. But instead of that, we are also interested about these gene expression marks, where we can see also the transcription factors that are primarily very important for the whole process of the metabolism. So, like I said before, we have different mechanisms how to influence basically of how strong will be the interaction between the proteins and the DNA. But for us, it's important that before the polymerase will start within the promoter region. So within the gene, we have some kind of a promoter region that actually tell us in which point this transcription of the gene should be started. And in order to be activated or in order to be expressed this kind of a gene, there is also influence from the enhancer that actually that enhancer is far away, could be far away from the part of the gene where that gene is expressed, which means that actually in this linear structure, that enhancer part of the gene could influence the interaction between the protein and the DNA on one side, but it could be far away. But in the 3D structure that we can observe within the DNA, basically it could be very close to this part of the DNA, so it can influence the gene expression directly. So what we have in traditional machine learning, first, The problem was that we have generated a lot of data in that point of time, high dimensional data, but we needed a good representation of that data in order to be able to learn something from the data that we have acquired. So we needed a manual feature engineering, high expertise in the domain that we should use in order to produce something that is meaningful, especially for the models that we would like to develop. And all the models that we have built were very specific for the task that we would like to solve, and also very biased to the data that we have generated. So that is not something that we would like to use in the future. We would like to completely change the landscape of the different models that we are going to use, and how to compare and how to use at least the data that have been generated in the past and are going to be generated in the future. So we would like to somehow address this topic from the multi-omics perspective. The data that are generated directly from the genomic, and transcriptomics, also proteomics and epigenetics, which means that from one side, we would like to explore the DNA structure. We would like to explore the mechanisms that directly are related to the interaction between the proteins and the DNA structure. But on the other side, we are also interested about what part of the DNA have been transcribed and what kind of RNA have been generated, mRNA have been generated, and that is part of the transcriptomics. And within the proteomics, we have the analysis of the proteins that have been generated by all the transcribed RNAs directly. So there are multiple fields that we can combine together in order to get a holistic view of the whole domain that we are trying to analyze and inspect. So what are the foundation models? Basically, the foundation models are the most general category within the generative AI, the new movement that we have seen within the natural language processing. So we are not pioneering in that side, but at least from what we have learned from natural language processing, we are trying to somehow accommodate and use the knowledge that we have obtained in the natural language processing, just to be able to apply the specific architectures of models within the domain that we are interested in right now. In order to be able to do that, we need to have basically two kinds of strategies for learning those models. The first one is the pre-training strategy of the model, and the second one is the fine-tuning of the model to the specific tasks that we are interested in. So I'm going to share some details about the pre-training process, but also for the fine-tuning process of the foundations model that are developed recently in this domain. So what is the approach? We would like to take all the data that are generated from the specific type or multiple different specific types, from multimodalities, from multiple different sources, but also from the same type or limited to the same type. And we would like to digest that data in order to be able to acquire some additional knowledge patterns that will be used in the fine-tuning process of the model. And one strategy that we are using here is the self-supervised learning, which enables learning rich representation without labeled data, which means that if we have some kind of genetic sequences, DNA sequences, we would like to at least learn some kind of a pattern that exists within that sequence without any additional labels related to the mutations, exist, not existent mutation, what kind of mutation we have within the DNA sequence. So what we are going to do with that? So basically, we are going to start the usage of the primary technologies that we have seen in natural language processing, that is mask language modeling, and also the next token prediction strategy, which means that without labeling of the data, we are able to digest and learn something from that data that is currently present. What are the advantages of the foundation model? First is the transfer learning. Models pre-trained on large dataset can be fine-tuned with small omics, single omics or multi-omic dataset that will improve the performance because we have grabbed from the data that we have learned the general knowledge there. We don't need a really specific feature engineering, which means that they have reduced feature engineering because they can automatically learn the representation, reducing the need of manual pre-processing of the data, which is quite important because our expertise, for example, for some use cases could be not sufficient just to be able to represent the problem in the specific way that could be beneficial for the specific model. They can make a better generalization. They are more scalable, and they can perform some kind of multimodal integration. So different type of data could be used, for example, for learning and building those foundation model, like in natural language processing from audio signals, from video signals, and also from text. And also here we offer some kind of additional interpretability based on some kind of attention maps regarding the features that attribute or contribute most to the decision that we are going to make, especially within the fine-tuning phase. So what we have basically within the foundation model, I'm going to explain this process as a part of the genetic or the DNA sequence, but it's basically the same for different types of data that we can have here, because everything is about some kind of a linear location, and for that location we are assigning some specific label within the data that we are playing with. So what we have, it's basically the DNA sequence, for example, and if we would like to somehow learn from general knowledge from the DNA sequence, we need to somehow give that DNA sequence directly to the model. We have some kind of a context size, like in the large language models, and the only thing that we need to perform is to make the tokenization of this sequence of nucleotides that we are observing. Just imagine within a natural language, for example, in one language, we can have multiple thousands of different words within the language. But here, within this language, the DNA sequence language, we have only four different nucleotides that could be combined in some bigger chunks, for example. Those chunks that I'm mentioning here is some kind of a K-mer grouping of these nucleotides in order to be able somehow to represent the combination of different nucleotides next to each other. And in this process have been experimented with different number of tokens. The first one is treating every separate nucleotide as a separate token. Then we continue, for example, with two of those nucleotides to be combined and grouped in one particular token, or even go to up to 8 different nucleotides to be combined and grouped within one token in order to be able to see the performance that we can achieve within those models. And based on the experiments that have been performed, around 6 different nucleotides combined within one token, we have at least some of the foundation model achieve the best results within the representation of DNA sequences here. Just imagine that every particular sequence that we can observe here is always related to some position within the chromosome, and for example, within the human genome or any other species genome. So what it means? That means that actually once we perform the tokenization of the problem of the input that we have, the DNA sequence, we perform the tokenization of the DNA sequence, and we can start with the pre-training of the model. So what we have as an output within within this model pre-training, we have basically 2 strategies that are mainly used: the masked language modeling that I'm going to explain, and also the next token prediction. But once we perform this kind of a pre-training of the model, we should proceed with the model fine-tuning with small subset of examples that will tell us that should give us more insights regarding some specific task that we are interested in. For example, give me a prediction of the mutations that have been seen within the DNA sequence, or give me some kind of information where the enhancer starts or where the promoter starts within the DNA sequence. Just based on the sequence that have been used as an input, we need to tag or label the sequence. So once we pre-train the model, we proceed with the model fine-tuning, and within the model fine-tuning, we have the same input sequences, but also on the output side, we have some kind of labels that will improve our behavior or the behavior of the model within the fine-tuning phase of the model. And that model fine-tuning could be quite different. For example, we can fine-tune only the parameters that are part of those of the model. We can fine-tune only some specific layers, for example, within the model, or we can just freeze all of the parameters that are part of this model and make some additional prompt fine-tuning that could be artificially added, for example, here on the input side, like it is presented here. So we have frozen models, so we are not learning, we are not tuning the parameters of the model, but we are tuning additional prompts that could basically give us a hint how to make a better prediction for the particular task that we are interested in. So what is mask language modeling or mask sequence modeling, the pre-training strategy that is used here? For example, we have some kind of a sequence of words. I am a virtual assistant. So we are trying to mask some particular word within the sequence of words. And in the output, within the learning phase of the pre-training phase of the model, we are trying to make a guess or make a prediction what is the most probable word to be seen in this part of the sentence. So in the main iteration that we are making, we are trying to guess basically the masked words that we have based on the surrounding context that we have. So in terms of the genetic sequence, DNA sequence, what we do, actually we mask some specific part of the DNA sequence, and based on the surrounding of that part of the DNA sequence, we would like to make a guess or make a prediction what is the part that is missing here. So that is the pre-training process. So we are generalizing and we are learning about the patterns that exist within the specific DNA sequencing and specific locations within the DNA. And there are different type of mask sequence modeling here regarding the tokenization that I have mentioned, and I'm not going to into the details into explaining this one, but there are multiple different slightly different strategies that are combined with the special tokenization and learning the model here. The next pre-training strategy that is used basically in this domain is causal language modeling or next token prediction, which means that we know about the first part or the left part of the sequence. We would like to predict the next token. The next token could be represented by only one nucleotide or multiple different nucleotides or tokens that are generated by some kind of a pre-processing step that could be byte encoding, etc., etc. It could be different. But the thing is that based on some specific sequence, we would like to predict about the probability what will be the next token, what will be the next token in the sequence that should accompany the previous sequence that is given to the model. How is that could be seen? For example, we have the sequence as an input and we would like to predict the next token. Once the next token is predicted, it is added to the input and it makes a new probability assign. Very similar like in the natural language processing, where in every point of time we are generating token by token. If we take that the token is a word, then in that case we are saying that we are generating word by word within the sentence until some kind of an end token is generated, which means that we need to end with the specific sentence or the specific answer here. But this is not the whole picture because we have mentioned only the genetic information. So the additional pre-training strategies could be seen within the epigenetics factor or even transcriptomics or even proteomics. But the most important one are mask methylation, which means that for the specific part of the genome, we have locations and we have measured some methylations, methylation across the CpG islands, which means we would like to know on average on that location, specific location, what is the level of methylated CG pairs basically within the DNA, which means that if we have high methylation of those CG pairs, it means that the gene cannot be expressed because the interaction between the proteins and also the DNA is tightened up. But if we have, for example, low methylation within the DNA, it means that actually our that interaction is relaxed and we can basically make a transcription of the specific part. And why this is very important, mask methylation and learning of foundation models with mask methylation, because the methylation, the epigenetics factors are mostly related to the detection of cancer, which means that by analyzing the profiles, the epigenetic profiles and the level of methylation that exists within the human genome, we can actually get a lot of insight regarding the potential of developing some cancer state within some cells within our organism. Or we can get some kind of a histone marks that are some kind of chemical modifications to histone proteins that affect chromatin structure and gene expression, of course. And each of these marks is somehow related to the level of gene expression. So we measure different factors, but actually they are all motivated and and all of them are related to what is the level of gene expression, in which part we will have some kind of a gene expression, and that gene will be able to transcript. Of course, if we go further on with some additional approaches of data generation, for example, we are not interested about observing the DNA sequence as a linear sequence of data. We can just go within the 3D structure, and these are high throughput chromosome chromatin capture, which are DNA fragments that are associated physically within the 3D space and give the interaction of the different parts of the DNA, how they can interact between each other. We can improve the quality of our models because we are adding one additional dimension of information that will give us a lot of insight how everything is structured within the chromatin, of course, and the chromosomes within the DNA. So what are the downstream tasks for fine-tuning the models? Usually we perform some kind of that is performed on already pre-labeled data, which means that we would like to predict whether some part of the genome is enhancer or not, for example. And we are dealing with the problem of giving the DNA sequence, and we would like to label the position where the enhancer starts or the promoter starts, for example. These are the downstream tasks that have been used for building this foundation model, or multiple of them have been used for fine-tuning the models, and we can find them, some of those foundation models available across the web. For example, some kind of epigenetic mark prediction, like methylation, hydroxymethylation patterns, or marks that could be visible and could be sensed within the DNA with specific sequencing technologies like Oxford Nanopore that is used right now, and also splice site prediction. So in which part of the DNA the exons and the introns basically are, and the introns are basically removed from that part of the sequence in order to be in order to build the mature RNA for transcribing that transcribes the gene directly. And for example, the slicer sites are usually related to some kind of a conserved motifs. It says that if the sequence starts with GT or AG, probably we are entering into a splice site. But actually, this rule-based system, for example, is related to many false positives because not all GT and AG are really related to the splice sites. But instead of that, we would like somehow to put a lot of DNA data within the model, and based on the conditions and based on the knowledge that the model acquired during the pre-training phase and during the fine-tuning phase, we would like just to directly specify which part of the DNA sequence is related to the splice sites. Regulatory element prediction, so the different types of regulatory, for example, like I said, enhancer prediction, or even methylation state, or even hydroxymethylation state, all the different regulatory mechanisms that we know that exist within the transcriptional gene of the genes, we are using for this fine-tuning of the foundation model. Chromatin profile prediction, etc., and etc. Spices classification, even just from the DNA sequence, just to know which kind of a species that specific DNA sequence comes from, etc. So what is the pioneer here? Which foundation model? The first one is a DNA BERT. Actually, it's a modified, slightly modified BERT architecture from the natural language processing, and it is learned on human reference genome. It's a transformer architecture, very similar to the BERT architecture, which is encoder only, and it counts around 110 millions of parameters. Just for a comparison, the smallest, I would say, not the smallest, but around the smallest capacity with a good capability of large language model that we are using today is around 8 billion models. So just imagine this kind of a model is much smaller. Even we have many different data and many different huge amount of data that could be learned. But instead of that, because of our limiting factor of how we express the knowledge about the DNA and the genetics and all the other multi-omics approaches, this model and the models that we are going to see as a foundation model will count up to, I don't know, 2.5 or 3 billion parameters. But most of them are much lower. So what are the downstream tasks for this DNA BERT that is used is the promoter region prediction, transcription factor binding site identification, functional genetic variant identification, enhancer prediction, genomic variant annotation, etc., etc. And what we have seen here that actually it predicts the promoter regions quite good, but also it can have a good accuracy for the donor and the acceptor accuracy within the splice sites. And also this DNA BERT demonstrates the ability to capture global and transferable understanding of genomic DNA sequencing, very similar to all the foundation models. There is an update. Last year there was an update for this model, and the only difference, apart from the small architectural difference of the DNA BERT, apart from using just the human genome data, for this model to be built, it was used also multi-species genome data. And the downstream tasks that have been used for fine-tuning this model are pretty the same as the one that the DNA BERT model was trained. Also, all of these two foundation models have been used the mask language modeling or mask sequence modeling approach for pre-training the foundation model. One of the most influential, I would say, today is the Groover foundation model. that counts around 1.2 billion parameters. And it was trained on eukaryotic sequence data and also genome sequence data and the human genome. The downstream tasks that have been used is the genomic function prediction, promoter identification, protein binding site identification, quite similar to those that DNA-BERT was used. Also, the interesting part here is, again, this is encoder-only architecture that uses the transformer blocks for building this model. But the interesting part here is that they used byte pair encoding, which means that before making the tokenization part of the model, they build a tokenizer. So based on the data that they have observed and they have seen, basically first they build a tokenizer that will be specialized for that type of data and then proceed with the training and the fine-tuning phase, which is opposite to what DNA-BERT and DNA-BERT2 made, taking into account just groups commerce of the DNA sequence. The next one, which is also very influential, and it appears like 4 months ago, it is nucleotide transformer, which is basically encoder-only architecture, mask language model, mask sequence modeling paradigm is used for building that one, but it does not use the commerce, but every particular token is basically every particular sequence, every particular nucleotide that we can see. And the results shows that they have a really good context-specific representation of the nucleotide sequence and achieved accurate prediction with limited task-specific data, which means that if you take this kind of a model and try to fine-tune with just a small subset of data that you have for one particular task that you would like to make and develop some kind of a model, this is a good choice because it generalizes well and it can be fine-tuned with a really small amount of data. The important thing is that it excels in epigenetics modification detection, which is quite important because, like I said, the epigenetic profiles is most related to the cancer state, cancer cells, for example, in the current research. Hyena DNA, it's another foundation model that actually take into account the Hyena CNN architecture, which is decoder-only architecture and deals with the next token prediction pre-training strategy. But the important thing here is that Hyena CNN is basically made just to overcome the limitations that we have within the context limit of the transformer architecture, which means that in this bioinformatics domain and genetics domain, it's quite important to have a huge context because, like I said, for example, the enhancers could influence quite a lot of how some genes are expressed within the DNA, but actually they could be far away, thousands and thousands nucleotides far away from the part of the DNA that is influenced by that enhancer. So this kind of a model can, for example, digest a context of 1 million tokens, which is something that was quite new in 2023 because we were not able to digest this kind of tokens in that point of time, especially in the bioinformatics domain, genetics domain. The other one is EVO, which is one of the largest model, which uses stripped Hyena, very similar to what we have seen previously. It is connected to next token prediction, and it has the biggest. It has the highest number of parameters, 7 billion parameters, and this one basically is very similar to what we have seen in natural language processing as the lowest models there with the smallest number of parameters. The interesting thing is that it can generate sequences exceeding 1 megabase and achieves competitive zero-shot performance on function prediction, gene function prediction. And it also incorporates the multimodal approach. The next one is the metal GPT, which is learned from the methylation mask within the genome, which means that we have the locations and we have about that location what is the level of methylation. So for the whole genome, we are learning this kind of a model, and it achieved very good results in understanding the methylation profiles of the different DNA sequences. CP-GPT is very similar to what we have seen within the metal GPT, and it is related to learning from the positions where we have CPG islands, which is quite important, like I said, especially for the downstream tasks that are related to the detection of cancer. The training data have been composed of 1,500 DNA methylation datasets, which is a huge demonstration of capability of this model. And it encompasses basically more than 100,000 samples from diverse tissues, which is quite important because the methylation patterns across the different tissues could be quite different. It outperforms specialized model in aging-related tasks. It is highly adaptable across different methylation platforms and tissue types, and achieves strong performance on biomarkers of aging challenge. It can identify CPG islands and chromatin states without any kind of supervision, like a zero-shot learning. Then we have the high-C foundation on that 3D reconstruction of the DNA that I have mentioned. It is CNN-based architecture, which is not basically part of this generative, I would say, generative models that I have mentioned previously. And because of that, it was important to add it here. And it is one and the only that I have found when I have made the research about the exploitation of this 3D genomic data that have been generated in the meanwhile. So what are the conclusions here? We have the explosion of biomedical data, and we would like to understand the global knowledge and to make a better and make a holistic view about what we have from what we sense from the genomic data. And because of that, the concept where we are going to generate some particular data and we are going to make some kind of a very specific model that will be highly biased towards the data that we have generated is not the way forward. So the way forward in bioinformatics is more related to the new concept, foundation models, pre-trained models that will be able to specialize very easy, that will grasp a lot of knowledge across the data sets that we have already generated and will be generated in the future. The important thing is that these models are interpretable, more scalable, and better equipped to enable real-world application in these areas. Thank you very much.
Thank you, Gjorgji, for your talk. And if our participants have any questions, they can use chat or Q&A sections, or they can directly unmute themselves and ask their questions. In the meantime, let me ask you some generic questions. I'm not an expert on this area, but if I say something wrong, please correct my question and me, okay? Lastly, you mentioned about models and their complexities. And foundation models often, as I understand correctly, often have millions, even billions of parameters, making them very complex and resource intensive. So in your opinion, how essential is access to supercomputing resources for training and experimenting with such large-scale models?
Yeah, that is very important, actually. We need to have in mind that actually those that are 100 million parameters is not something that is very huge. So, but if we go into the direction of billion models, in that case, we will need a huge, good GPU cluster in order to perform some kind of at least pre-training. But if we want to perform some kind of a fine-tuning, we can also manage that one with a small GPU cluster of 2-3 GPU cards. It will not be a problem. And usually what we are going to do is basically fine-tuning those models. There will be some centers, and of course here we can also do it. In our supercomputers, on our cluster, we can also perform some kind of a pre-training. If we cannot imagine on these areas, on this region, to perform pre-training on, for example, models, large language models in natural language processing, for specifically for this bioinformatics domain, genetics domain, we can perform, we can afford. With the new establishment of the AI factories, probably we will be able to perform this kind of even pre-training of these models. For fine-tuning, it will be much more easier, and we can make a big influence here if we want to make some additional research, because we are quite close in terms of large language models, because we do not have a proper resources to performing a pre-training or even fine-tuning bigger models.
Okay, thank you. About talking about some technical, maybe ethical barriers and some future-looking perspective. Artificial intelligence has shown great promise in analyzing complex genetic data. And in your opinion, what are the biggest challenges in applying AI to genetics today, and how can we overcome them?
That's a huge topic right now, especially how we can explain how we can regulate AI. Of course, there are ethical committees regarding, and it's getting even more difficult and more difficult in that case, because when you need to approve something, you cannot give a consent about everything. So you cannot just train the model for whatever reason. So you need to focus on some specific without understanding what is going on the other part. And we need to take an opportunity that AI will help a lot in detecting some specific diseases, in detecting which type of drugs could be investigated and how they can influence not only on the local small parts of the genome, but also across the whole genome, what could be the influence there. And this is something that we will see in the future, and we are going to fight with this. But the most important thing is that if we achieve some certain capacity of these models, it is much easier to claim that this is for good reasons that to be made.
Thank you, Gjorgji. Do we have any more comments or questions from our participants? If so,
may I have a question?
For sure.
Well, I'm a geneticist and a PhD candidate at Madjarov, and I'm working on ancient DNA. Ancient DNA data is very scarce, and I wonder about these models. How much data do we need to fine-tune these models?
Okay, so for example, to fine-tune this model, it depends quite a lot for how we are going to freeze some layers of the model. So in order to be able to fully fine-tune a model, we will need a lot of data, like, I don't know, 50,000 samples in order to fine-tune the full fine-tuning of the model. But even with
you mean genomes, whole genomes, like
not directly for the specific task that we are interested in. If we have one sample from one genome, for example, we could have, I don't know, thousands, hundreds of thousands of different samples. I'm talking about samples. So, for example, if you want to build some kind of a model or fine-tune a model from these foundation models, we can have from 100 or 200 person that will be able just to fine-tune. To pre-train, that is not possible. But for fine-tuning the model, probably we will not be able to fine-tune all the layers within the model, but just to fine-tune some specific parts of the model, or even to perform some kind of a prompt fine-tuning. Depends on the task that we would like to perform, and depends on the number of samples that we have actually.
Thank you.
Any other comments or questions? I guess no. And we would like to thank you, Gjorgji, again for your contribution and insightful presentation today. Thank you.
Thank you very much once again for this opportunity. It
Thank
was a
you.
pleasure to share my insights. Thank you very much.
Your presentation and the video recording of the seminar will be on our event page and our YouTube channel. And our next and last seminar under this joint organization coordination will take place in 2 weeks on Tuesday, July 22nd, titled as AI Meets Precision Agriculture. Please, our participants, do not forget to expand this to your colleagues who may be interested and register. We look forward to seeing you in the next session. Thank you all for joining us today and have a great day. Thank you.
Bye. Thank you.
Bye.